All right, here, eukaryotes are pretty straightforward. You have your mRNA. Here's your start, your AUG, or almost always an AUG. It goes along to get a stop. There are 3 stop codons. Let's say UAG, for example. So we've got a stop codon here, and you make a protein, and you're done. Nothing interesting. Prokaryotes. mRNA. I have my start. I make a protein. Sometimes I have another start. I make another protein. I told you it was the first AUG. Well, it turns out that in bacteria, you can actually start translating another protein later down the message. There's another place the ribosome will get there. It isn't really the first AUG in bacteria. I lied. Oh well. And it turns out you might have another one over here. And I make another protein. In fact, I can make multiple independent proteins off the same long message. This is called a polycistronic message. Cistronic sort of means on the same strand. Many things on the same strand. I can actually make 6 proteins off a single message. Why bother doing that? Suppose I wanted to make 6 proteins all at the same time. Give me an example why I might want to make 6 proteins all at the same time. They're all used in one process, one biochemical pathway. Maybe they're the 6 steps of a biochemical pathway. So in the genome of my bacteria, if I had 6 separate genes, each with their own regulatory controls to turn them on, I have to have DNA involved in making all that regulatory control. But I'm a bacteria and I'm competing by replicating my DNA, and I want my genome to be small. Why not just slam them together in front of one promoter, make one long message, and have all the proteins I'm going to need for this new metabolic process that I want to induce right now, this new pathway, on a single message? So bacteria, because they're conserving their DNA, because they don't want big genomes, will often string together a lot of proteins on a single message. Very efficient of their DNA. Just like you're profligate about your DNA with all these introns and things like that, the bacteria are pretty worried about it, so they'll just stick them all together and you get great coordinated regulation. Suppose you're a virus. Suppose you're a virus. Do you worry about how big your genome is? A lot. You really want to get every bang for the buck. So I might want to have messages that were polycistronic if I was a virus. But could I do any better than that? Yes. Could I overlap? What do you mean overlap proteins?
There's a certain amino acid chain that's in both proteins. That's the genes for them overlapping.
But I mean, I've got a certain reading frame. But you want me to, how could I get 2 different sequences out of exactly the same RNA overlapping? Shift the reading frame? So wait, let me see if I can get this right. You're proposing for a virus that sometimes you would like to have a segment of this RNA that gets read in this open reading frame, but also might get read in this open reading frame. Shifted one over, and you would make 2 different proteins. And you're even proposing, I guess, that if I was really clever, I have 3 reading frames available. I could start +0, I could start +1, and I could start +2. And I actually, in theory, you're proposing, could get 3 separate proteins out of exactly the same message if I really wanted to. Is a virus allowed to do that?
You wrote it on the board so promptly.
I wrote it on the board, which is a pretty good sign that it's allowed to do it. In fact, HIV has a segment of HIV that is translated in 3 separate reading frames. It is translated, there's a splice here, here, translated, this segment can be translated in the ONV protein, it can be translated here in REV, and it can be translated, I guess, TAT here. You can get it translated in 3 different reading frames. Pretty amazing condensation of information. So basically what I told you was you have a very simple central dogma. DNA goes to RNA goes to protein, you replicate your DNA, you copy your DNA into RNA, your RNA gets translated into protein, it's all very simple. Life never leaves anything alone. Once you have something simple, it starts making it more complicated. It starts saying, how can I optimize it? I'm competing, it's all competition down there. The bacteria are competing, you're trying to innovate, you're trying to do things. And the innovations happen at many different levels. There are innovations about what kind of genomes you can have. And viruses exploit all those different kinds of genomes. There are innovations about how you can pack information in genomes to make messages by splicing them up in all different ways and getting creative molecules in your own body by having alternative splicing, and creation and evolution by having exon shuffling to create new proteins. You can pack multiple proteins into a message. You can even pack multiple proteins right on top of each other into the same nucleotides. It's a pretty impressive thing, and it's a good rule that once you've got a handle on something simple in biology, you can count on the fact that biology is constantly tweaking, constantly optimizing. It's a pretty amazing system. All right, that's all for now. Okay, stop for a moment and test your understanding with this question about translation in different organisms.